Thank you. So thank you, Sukhvinder, and hi, everyone. I thank you, PacBio, for inviting me to speak here. We're completely new to the long-read sequencing field, and PacBio has made it really easy to adapt and move over from short reads. And I'll tell you a little bit about what we've been doing over the past year and our 10,000 genome project at the Estonian Biobank. So disclosures, PacBio, Illumina, and ONT have all provided some kind of reagents and allowed us to test their different platforms, and I'll tell you about that experiment at the beginning of my talk. So our main project that is focusing on these 10,000 genomes has been to figure out how we can use or create the best possible imputation reference panel and use these individual genomes for looking into new genetic findings that are clinically relevant in the broader population. And the idea of this Team Per Med, which is an European Union plus Estonian government investment of 30 million euros, like 15 each, it has allowed us to build a center for personalized medicine in Estonia that brings together the university, all the researchers, and the hospital to figure out how we could best implement everything that we find in the research arena. So the idea is to not only make discoveries in genomics and publish papers, but to also look into cost-effectiveness analysis and create new treatment or guidelines for medical staff of how they could use all this genomic information and bring the payers, who are then supposed to pay for all these new services in the healthcare setting. So it has a lot of different partners, and the international partners are then EMC Rotterdam and FIM and the University of Helsinki, a bit broader as well. And we really hope to deliver, and one big part of this project was to do genome sequencing for 10,000 individuals from the Estonian Biobank and to create or capture the genetic diversity in the country. And those who haven't heard of the Estonian Biobank, we have 212,000 participants, which is 20% of the adult population in Estonia, and all have been genotypes, and we have Nightingale metabolomics data for all. So far, we had about 3,000 short-read genomes and exomes, and for all individuals, we have consent to annually or regularly update the health data or phenotype data from the electronic health records in Estonia, which are all deposited into one central e-health database, both primary care, secondary care, and all prescriptions, procedures, and everything that's going on in the healthcare system. We have a single-payer health insurance. So based on this data, we're doing a lot of research and discoveries, and if you're interested more about the work, you can scan the QR code to see our recent paper that really gives an overview of the whole biobank and the research we've done, and how we're trying to get this information to implementing personalized medicine in Estonia. So these 10,000 genomes that we selected for this Team Per Med project, they are supposed to capture the genetic diversity, as I mentioned. And it's a small country, so 10,000 genomes will take us really far. And so since all of these individuals have been genotyped, we wanted to create the best possible imputation reference panel to study all of these variants in the rest of the biobank. And then we will also do recall studies based on the clinical findings, whether it's the American College of Medical Genetics recommendations for secondary findings that are actionable, or broader into looking into pharmacogenetic variants. And personally, I am extremely interested in pharmacogenetic research, and we've also done recall studies of individuals with novel findings in drug metabolizing enzymes to figure out their actual effect in real people. And since we want to do both return of results and research and capture the major genetic diversity, our initial plan and the cost of sequencing back then when we wrote the grant was to do 9,000 short-read genomes and 1,000 long-read genomes, and then use these long-read genomes to better phase the haplotypes and get a bit further with all the reference panel putting it together. And we set out then to compare 3 long-read sequencing platforms, which was back then PacBio, ONT, and Illumina complete long reads. So this was a joint collaboration. At a later stage, we brought in some colleagues from Uppsala. So Adam and Lars have been advising us a bit on the long-read sequencing side, which was new for us. And the rationale for testing the technologies was that most publicly available comparisons have been done on cell lines, and we didn't really know about what about biobank samples that have been stored for 10 to 20 years, and how do these platforms perform on these. And in many of these comparisons, the companies remove the difficult-to-sequence regions, which we do not want to miss. And we also wanted to test the workflow in the lab. If you're learning like 1,000 samples, how user-friendly are these different platforms and the bioinformatic tools as well. And since there's a lot of data going created, we also wanted to look into the storage and compute costs for processing all these genomes. So we did small pilots with all 3 platforms, and there was an overlap of 8 samples across all platforms. And the Illumina complete long reads are then actually synthetic long reads based on marked short reads. So some people are calling them complete long reads, some people are not. But then we compared the detection of SNVs, our single nucleotide variants, and here you can see that there is a great overlap and all are performing fairly well. We've got over 3.7 million SNVs that are detected on all 3 platforms, and there is really just small bits that each platform is missing. And in this slide, you can see that even if we look into the ones that are only detected by 2 platforms, PacBio is outperforming the other ones clearly. But the biggest difference was actually when we looked into indels, and here you can see that there is great overlap between all 3 platforms, but it's also about 220,000 indels that are missed by ONT. And they may tell you that these are not important, but we looked into these variants as well, and we found that even in our 8 samples, there were variants that were in ClinVar. They were either, most of them were called unknown or conflicting reports, while there was even one pathogenic one in these 8 samples. So the next step was to compare detection of structural variants, which was a lot more complicated and which has taken us quite a bit of time to compare the overlap and how do we define the same SNV in 3 different data sets. And here is a simple comparison where we expected, I think this is Truvari, so both looking at the start and end of the sites and defining it by at least 50% overlap of the SV. And here it's quite interesting to see that the Illumina complete long reads are actually missing quite a big fraction of the structural variants. Or it's the bioinformatics. We don't really know where this difference comes in. But in all of these comparisons, you can see that PacBio stands strongest. Since we're interested in pharmacogenetics, we also did a comparison of how well these different platforms can capture CYP2D6, which is the major drug metabolizing enzyme that metabolizes about 20% of drugs. And it's a complicated gene. It has duplications, deletions, hybrid genes with CYP2D7. And it's quite difficult to distinguish these from each other if you have short reads or array data. So we selected a few complicated samples where we could see from the array data or short-read genomes that something is fishy here. And for example, this is an individual who has a deletion on one allele, that's the SAR5, and has 2 copies of a hybrid gene followed by a normal copy of a non-functional STAR4 allele. So if you're into pharmacogenetics, this is incredibly complicated, and so that basically this individual has no metabolic function at all, because the hybrid gene is non-functional, star 4 is non-functional, and the deletion is non-functional. So the problem here with the short reads is that none of the pharmacogenetic star allele callers could call this diplotype. And for PacBio, the long-read genomes, both Pango and star phase, got it right. We don't know if there's 2 or 3 copies of the hybrid gene, but that doesn't really matter. There's a lot of reads, and if you're looking into the reads, you can see that where you see all of these mismatches is where the hybrid gene or misalignment occurs. And these super long reads can actually show you that there's one hybrid gene here and another hybrid gene there, so they are in tandem for sure. So there's at least 2 copies of the hybrid gene. And ONT hasn't really built their pharmacogenetic caller yet, so we tried the existing tools, Pango and ALDI, and ALDI couldn't call this diplotype, while Pango, which is PacBio developed, actually can pick up, kind of gets some of the alleles right, but can't place them in order, while Pango could do it on the PacBio data. So in summary, in this comparison, we found that there was similar performance across all 3 platforms for SNVs, while for indels, ONT clearly has difficulties and missed about 6% of the variance. One positive note on ONT was that the lab found that actually running ONT in the lab was smooth and easy. And of course, the instrument costs are extremely low compared to the other platforms. But what really made a difference was also the bioinformatics. I think we have 25 pages of email exchange with ONT bioinformatics support to actually get the tools to work in our lab. And I think there was a lot of cursing in the bioinformatics room as well. And we also find that the ONT files were 10 times larger, which definitely influences your storage and compute costs. You might be told to delete some intermediate files, but there's nothing in the instructions or protocols about that. And ICLR did not perform as well as we hoped, and is also extremely expensive, so that was also off the list. Now Illumina is working on a new constellation tool that we're also testing and seeing if that might work. It should be a bit cheaper. And another advantage is that ONT and PacBio generate full methylomes, which is definitely extremely exciting. And it requires more DNA, but that's not an issue for us. So based on these tests and the support that we got and the different offers that we got from the different platforms, we were actually able to redesign our whole project to do 10,000 HiFi long-read genomes. And we are not interested in short reads anymore. So now we have 3 Revios in the lab working day and night. We have an Agilent femtopulse and just recently purchased this Hamilton robot for sample prep. It has not been launched yet, so we're waiting for the training to start using it. But we also have this A-team of people who are running all these different processes. So Steven, who is here in the audience, you can find him if you're interested in the DNA and automation and how they have set up everything in the lab to run the samples, together with Tuuli, who is also here in the audience, head of the sequencing lab and in charge of all these processes. And Georgi, who couldn't make it, is the one who has set up the whole bioinformatic pipeline. So the goal is to get 20x long-read genomes for 10,000 Estonian Biobank samples within 2 years. And we got the instruments at the end of Q1 in 2024 and started sequencing beginning of summer. And they worked through the whole summer holidays. And by the Q3, we reached maximum throughput and got the yield optimized for version 1 reagents, and then this beginning of this year we then transitioned to Spark, and you'll see how this process has been going. And today we have completed 2,000 samples at 20x. So I won't go through all these details. If you're interested in how everything is set up in the lab, you can speak to Steven and Tuuli. but basically the only step that so far has been done in an automated fashion is the sharing with a 96-headed robot, and the rest has been done manually by 2 lab technicians. And this is then showing the workflow for version 1 reagents and for Spark. And here you can see that they have switched to creating 12-plex pools to 96-plex screening pool, which is first tested on 8 SMRT cells, and then everything is repooled to optimize the yield for each sample. And then they are sequencing the 96-plex pool on 40 SMRT cells. So if there's a problem with a SMRT cell, we'll see that immediately. And then the yield we are aiming for is 60 gigabases per sample to get around 20x coverage. And the final yield, we see a variance between 5 to 10% between the samples in a 96-plex pools. And you can see that I'll show the median output yield as well on the next slide. So here you can see the median output that we're getting, or yield, for the version 1 reagents was around 114 gigabases, 95 gigabases, while for the Spark it's 114, and our record right now is 141 gigabases for a sample, and we're trying to get that even further. So here you can see the whole picture of all our runs and the yield that we're getting per SMRT cell. And the major variation that you can see in the light pink is basically different lab technicians, which we hope now automation will remove. And then we reached a very good platform or optimization at the end with version 1 reagents, and then came Spark, initially doing well, and then a small dip, and trying to optimize it even further now. So far, we have completed library prep for over 3,000, close to 4,000 samples, and initial sequencing is completed for 3,200 samples, and the total yield is about 210,000 gigabases, and now we are at a speed of 144 genomes per week. So the bioinformatics, I have a few slides, I'll just show you them, but if you're interested, you can speak to Georgi about it, just email us and we'll connect you. So he first of all rewrote the PacBio Crumble pipeline in SnakeMake, since this was his preferred language, and also optimized it for the Slurm HPC cluster that we have. And the major steps that this entails are then alignment, variant calling, and annotation. And he also optimized the memory allocation for each step, and then used benchmarking. So this is the whole process, and this is then the benchmarked data. So if you would like these tables, we're happy to share them. But then it really shows the optimized amount of memory allocated and the time each step takes as such. And he also compared this for PBSV and Sawfish in comparison, just to see the computes and switching from one to another. So the sequencing metrics for now, it's that we are getting a median coverage of 21.5x, and the median read length right now is 14,000 base pairs, and yeah, I need to move faster. So a sneak peek into the first 2,000 genomes for pharmacogenomics. Whoever is interested in CYP2D6 and CYP2C19, we're showing that we have found about 193 different alleles, and you can see that what kind of stands out on this image is that these hybrid genes are much more common than we thought, and I think previously we have been getting them miscalled as duplications, while they are not. And we've even seen, you can see that where the hybrid gene starts, in which exon of CYP2D6, and then switching to CYP2D7 can also vary within the star 68, which is the hybrid gene. And an important point is that this star 68 has always been seen in tandem with star 4, And now we have started to see this star 68 when we're getting to 2,000 samples. We've already seen a few examples, already 4 samples, where star 68 is actually followed by a star 35, and who's into pharmacogenetics will know that although star 4 was a non-functional one, star 35 is actually functional variant. So if you're assuming that star 68 is always followed by star 4, you will miscall this as a non-functional allele, while a star 35 should actually be functional. So any bioinformatic pipeline that is making this assumption will get these individuals or genotypes wrong. And whoever knows Andrea Gadik is a big person in pharmacogenetics fields, and she didn't believe us that we're seeing star 68 with star 35. And since we only had 20x genomes, we only had 2 reads that confirmed this tandem, she didn't believe us. So we sequenced this sample even further at further depth, and now we have much more reads confirming this and much more individuals confirming this. So we're very confident that this is actually correct. And for ZYP2C19, the picture is easier. There's not that many star alleles, although the gene is much bigger. But what we have seen here is a deletion of one allele that is present in 2% of our samples. And this was something that PB star phase was not calling. But I think the bioinformatics support from PacBio has been amazing. Thank you, Mike, and the whole team. So when we told Matt Holt, who is the author of PB star phase, that we have this deletion in the ZYP2C19 in 2% of the samples, a week later, he had rewritten PB star phase to call this deletion. So this is the kind of collaboration that we love, and the kind of support that we're amazed by. Now we're getting star 38, star 37 called in all these ZYP2C19 genes. So I really wanted to give you a bigger sneak peek into these 2,000 genomes and structural variants, which is definitely a very interesting field and has huge impact on the clinical outcome. But instead, I show you this slide. We're in desperate need of more bioinformaticians to actually process all these amazing long-read genomes. And if you know anybody, or if you're interested in working with us on these 10,000 long-read genomes, please contact me. However, we managed to look into at least the ACMG list of genes and try to find something interesting to show you today. And it is a bit difficult to Titin gene. Is it real? No, it's huge. There's so much in all of these genes that we try to annotate that we then wanted to find coding structural variants in these genes. And at least one interesting deletion that we found in BRCA1, which overlaps with exon 3, is a 5,000 base pair deletion in BRCA1, and we couldn't find it in GNOMAD. But when I spoke to the head of the clinic, at least they have seen it once in a clinical sample. So it must be extremely rare, but these are exciting to pick up. So next steps, we will keep sequencing, and we're really excited about calling all the genetic variants. We will phase them and impute them into all the 210,000 biobank samples, so we can then test all these new variants versus different phenotypes, whether quantitative or just electronic health records. And we're also very excited about the epigenomes and epigenetic clocks based on whole genome sequencing data. So I just want to say special thanks to the entire PacBio team that has been supporting us through this process and made the transition to long-read sequencing so smooth and so exciting. And especially the bioinformatics team has been of extremely great help and support and very responsive. And these are the acknowledgements of our own team and the funding agency from the European Commission and the Estonian government that has allowed us to take on this huge project. Thank you.